to see this movie later on and to follow up on that what is being discussed today. We have Tiziana over. Tiziana is working with us as a now, I'm sure also with Brock, together in different areas. We are really happy to have her here because she's a real capacity. So I want to introduce you a little bit further. Tiziana got her master's degree in industrial biotechnology in Bologna, Italy. She got her Ph.D. in Oenology and Agriculture at Padua in Italy, and was involved in a selection program for new yeast for cold climate winemaking. She also worked in Montpellier and Dijon in France, in which she came in contact for the first time with Brettanomyces, which is obviously her seminar today. And then in 2011, Tiziana joined Scott Laboratories and Lallemand, the Lallemand team in Italy. And her job there is technical support for Italy, and also she's taking care of R&D in Italy. We will have 3 seminars, so to speak. We want to put a lot of information in this session. First seminar will be over Brettanomyces, and then we have 2 other following this one. First one will be approximately 30 minutes, the other ones will be 15 minutes total. So we want to start right away, not wasting further time, and we will have a short time afterwards so that we will lead the discussion and we can ask questions. Let's get started.
Good morning, everybody. I'm really pleased to be here, and I want first to thank Brock University and Scott Lab for inviting me. And I will start from Brettanomyces, which is, I hope, interesting to you, since it's a global issue for enologists all around the world and for consumers as well. I will start with a few shots about how bread is perceived, just to let you know that also the wine specialized press is kind of interested in Brettanomyces and also in the genetics beyond it. Here another slide just to show probably the proportion of the problem. This is this data comes from the International Wine Challenge in London, and you can see that 10 to 15% of the total falls in this competition come from bread. Bread is currently considered the major microbial cause of wine spoilage, and you have to consider that these wines are sent from the winemakers to the competition, so they're not supposed to be bad samples of their wines. So about Saccharomyces and Brettanomyces, we know that both microorganisms are well adapted to the winemaking environment, and that both probably exist from the vineyard to the winemaking. But they take place at with different timings. When the grape is crushed and in high sugar environment like grape must, Brettanomyces doesn't grow. It's Saccharomyces that grows and performs the alcoholic fermentation. Then later, when sugars are down and there's few sugars, high alcohol, there's the right time for Brettanomyces to develop. So how does it spoil wine? We associate the bread character with some compounds. There are actually a lot of compounds, but the major cause of wine spoilage is the production of ethyl phenols. This production comes from a double reaction, so it's 2 enzymes involved, a decarboxylation and a reduction. The first step is not only Brettanomyces that can do it. We can also have some Saccharomyces strains, usually not the selected strains, but other wild yeasts. The second one is only catalyzed by an enzyme from Brettanomyces. So this is why we associate all these compounds to the bread character. So about its physiology, we know that, and we expect that some features of Brettanomyces are really similar to those of Saccharomyces, because both yeasts are really well adapted to winemaking. Indeed, they are both able to ferment. They can have an anaerobic metabolism, and which is more important, they are both ethanol tolerant and sulfite resistant. Then it depends on the strains for sure, but in general, both species are ethanol tolerant to high levels and sulfite resistant. But there are also some characteristics of bread that are different from Saccharomyces, and this is expected again because it has a different timing in winemaking. Indeed, bread doesn't like the environment where sugars are highly concentrated, like the grape must. And on the other hand, on the contrary, it can assimilate alternative carbon sources. And this is very important because it can grow on sugars other than glucose and fructose. There's not only this, and it can metabolize ethanol in some cases. So this is something really peculiar of Brettanomyces. Moreover, this is something really new. It has been demonstrated recently. It was, let's say, in the air for many years because some people already observed this. It seems that sulfite resistance in bread, it has a different mechanism than in Saccharomyces because it goes through a viable but non-culturable state, which is quite outstanding because it was known only for bacteria, and this is the first eukaryote, I'm sorry for the pronunciation, that shows a VBNC state. So,
sorry, what is VB?
Viable but non-culturable. Do I spell it correctly? Okay, I will explain later what's the meaning of this a little bit, and it's a kind of dormant state which is in between dead and alive. I will say this is not really scientific, but so what happens in winemaking? Bread is very efficient in colonizing the habitat, so because it needs few nutrients, so it can stay for long periods in your winery. But then, when during alcoholic fermentation, it doesn't grow that much. Saccharomyces dominates, even if bread is there. Later, when low sugars are available, this is a very small amount, and it includes non-glucose and fructose sugars, and alcohol is higher, the selection favors bread, and this is why bread comes up later. So, again, about the wine spoilage, I will be quick on this. Bread is not only dangerous because it produces the volatile phenols, it also can produce acetic acid in high amounts if there's oxygen. These compounds, tetrahydropyridines, that are responsible for the mousy smell, which is associated to bread. So this is not ethyl phenols, and for sure the ethyl phenols that are the major cause of wine spoilage. About this production, this wine spoilage, there are some critical points that are still under study and they are not clear. For example, we know that sometimes small contamination are enough, and this means that we cannot tell which is the minimal population of bread to have the wine spoilage. Sometimes wine spoilage starts with low population, sometimes with high population, and it's not clear why. What is the difference? Again, we know that 1 on 6 bread strains do not produce volatile phenols, and we don't know why. What's the difference between these strains and the other? And again, this ability to survive in the viable but non-culturable state, the mechanism we don't know what is the mechanism underlying it. So what is new and will help all the research on bread is that really recently the whole genome sequencing has been closed. The data about the genome are not available yet because they're doing the assembly of all this data. But in the Australian Wine Research Institute, they got the genome after 20 years of research. So this will be a big change for people who work on Brettanomyces. Because so far we only had 40% of the genome sequenced, some shots, but not assembled. So it was kind of difficult to work on bread. We just took advantage, the researcher, of this similarity to Cerevisiae and had some work. Now we know that among its genes, there are some very interesting ones. For example, the two encoding for the two enzymatic activities that go from the phenolic acids to the ethylphenols. We know that among this genome there will be the stress-responsive genes that can explain why bread survives in wine. So these physiological characteristics. And also, in the genome, it will be very interesting to find, probably one day, the genes encoding for the entry and exit to the BBNC status. So what is known so far about the volatile phenol production, the genes are already known, and also the conditions for these enzymes to work are already known. These have been found before the genome sequencing with specific experiments. So these two enzymes are pretty well characterized in terms of activity, at which pH or temperature. We can find data for this. What is not known is when and why these genes are switched on and switched off. And this probably can come after the genome sequences, because probably we will learn where they are in the whole genome, how they are managed by the yeast. So we will have more information. Again, about the adaptation and stress response, some data are already available. This is a fermentation where Brettanomyces, which doesn't grow that much, as I previously mentioned, and Saccharomyces were fermenting together in an experimental synthetic mast, and their stress response has been measured throughout fermentation. And what was previously known and also observed in this experiment is that Saccharomyces has a general stress response. It activates some proteins that are useful to counteract stress, and usually Saccharomyces turns on these genes around mid-fermentation. So when alcohol gets to, depends for maybe 5% more or less, and the cells are entering the stationary phase, Saccharomyces turns on this response to get ready for the later conditions. And bread, it also has something similar, which was not known, and so it has a kind of general stress response, it seems. This is kind of similar to Saccharomyces, but if you look at the timing, it's activated much later. So this seems to be conserved, but with a different timing, which will be consistent with the fact that bread is better fitted for later stages of fermentation. And again, a lot of other studies will come, because once you have the genome, you can search on many, many genes what's going on during the fermentation and at the end of the fermentation. Again, here there's the viable but non-culturable state. This is, you see, it's kind of a couple of weeks ago published. And here they have the bread population with, this is the viable population after a sulfide stress measured by cell cytometry, flux cytometry, I think. And so these are the cells that are alive, but if you put them on a petri dish, you get 0. So they can't grow on a petri dish, but they are still alive. And then when you remove the stress, the sulfite is removed, they can grow again. And this can explain why sometimes bread problems seem to be over in wineries. The problem is not turning out and for a while, and it can be a very long period, and then it starts again with no contamination from outside. It seems that when sulfite lowers, the sulfite level lowers, bread can start again. And so this could explain some issues with bread in winemaking. And this also can explain why sulfite sometimes is not enough to fight bread. So again, we will have new information. This is really recent. So, the post-genomic era for bread is coming soon, but what now? Winemaking is now. So, people have been fighting bread for a long time, and it works. So, how can we do? First of all, hygiene. I will not go very much into details. Just one take-home message: it's not only the barrels. We do know that barrels are the most friendly environment for bread. Bread has been found also in the pavement, in the ground floor, in the wineries, or in some niches, also in the steel tanks. So just be careful, it's not only barrels. Again, a good alcoholic fermentation management prevent sluggish fermentation and prevent malic sluggish fermentation. Every time you have your wine without a good population of microorganisms growing on it, no growth or slow growth, and no sulfite again, because you have the malolactic to be done, so you cannot sulfite. You leave some periods where bread can acclimatize well on your wine and can grow. Probably it will not rise that much, but it's ready to grow again after the end of fermentation. So this is some management preventive ideas. And here you can have some data. I will be quick. Here you have a quick malolactic fermentation. This is malic acid. There was a bread contamination, so bread started to raise, but as soon as the malic acid was consumed, acidity was added. So bread population was kind of under control. In this case, sluggish malolactic fermentation, they could add acidity only here and look at the bread population. So same case, more time for bread, higher population. And here you have another result. I'm sorry, there's some Italian left. This is co-inoculation, the brown bar, and this is spontaneous malolactic fermentation. So with co-inoculation, you can stabilize your wines much earlier, and this is the result for bread population and volatile phenols concentration. These are some French studies. But sometimes this is not enough. Good hygiene in the winery, good management of fermentation, and you still have bread. It happens. And this is an explanation. This is a average sulfite addition after the fermentation. So if you have some bread, you can have no culturable, but some viable bread, again, when you add sulfite. If you happen to have sulfite-resistant strains, and bread is a species, it has a lot of different strains, and you don't know what's in your winery. So if we happen to have sulfite-resistant strains, with sulfite addition we only achieve this, viable but not culturable state. This is a risk. So how to solve this point? Because there are some curative methods proposed. It's just a review. What I want to say is that you don't have curative methods for both the volatile phenols and the bread cells at the same time. And it makes sense. One is the removal of a chemical volatile molecule. The other one is fighting in a live cell. So it's two different goals you want. For the volatile phenols, the main idea is adsorption. So there's nothing specific. You have several tools that are being shown to have a partial absorption of volatile phenols. Some of these are lab scale trials, so just take the message and find out if for regular winemaking and your case of winemaking this works, because sometimes it depends on the pH of the wine, depends on several other conditions. For example, this is really new, and so far this only works with stirring. So let's give the chance to this method to see if it works in a real winemaking environment as well. The same for fighting bread cells. So filtration is a good tool. The only little bit tricky point is that it seems that the VBNC cells have a smaller size than the others, and so they can pass through the filter sometimes. This can explain how they found bread in some very old bottles that had been filtered 15 years ago, but they're bread now rising. So it's unlikely to happen, but it's possible. Then there are some unspecific treatments that can help a little bit. The temperature, so the flash pasteurization has been shown to be pretty effective with bread. It depends on, but it has an impact on the wine aroma. So in Europe, people are not really convinced about this tool, but it's pretty effective. And again, the dimethyl dicarbonate is also effective at quite high doses, but these are allowed in the United States. On the contrary, the DMDC is not allowed in Europe, and as far as I know, in Canada, neither. So this is another tool that exists, but for the European people, that's not really interesting so far. So how to solve? Again, we have some results from a new tool to fight Brettanomyces. This is chitosan from fungal origin. The chitosan is a polysaccharide. It comes from the same family of the cellulose, starch, and chitin. And in this case, doesn't come from jellyfish, but from Aspergillus niger, a completely safe microorganism. And through the chitin, you get this polysaccharide, the chitosan. And chitosan is already used in a lot of industries, including health, food industry for the smart packaging of some foods. And so it's now getting more and more approved for enology. In Europe, it's already done. In the US, it's halfway done, let's say. And so it's coming there. I don't know for Canada, but it seems it will come soon also. So these were the first results for the first chitosan application. And this was a lab scale trial during a Ph.D. work in France. And it seemed that from 2 grams per hectoliter, it's very effective from a pretty high concentration of bread cells at the beginning. I have to say that all the methods for fighting the bread alive cells, they are always dependent on your initial population. Because if you have 99% of good result and you have 1 million cells at the beginning, 99% means you still have 10,000 cells alive. If you have 10,000, you will have 100 cells alive. So everything is dependent on your initial population. These were some trials in wineries in France. So it's a couple of years that it has been tested, and to date the dose of 4 grams per hectoliters and 10 to 15 days treatment is what we suggest. But new data will be available this year to extend possibly this treatment time. And here again you have just an idea of the 4 grams per hectoliters treatment. This was the control. And this is just to make sure, here the plate count, so this is the number, And here is the real-time PCR, and it's pretty low. This is just to make sure we don't have only V, B, and C, because the real-time PCR will detect the V, B, and C state. So if there's nothing here, there's nothing in V, B, and C state as well. Okay. So far, but new results will come because the V, B, and C state has been shown very recently. Another trial, this was in Spain. This was done also winery scale. This was the population, so pretty high. After the 10 days treatment, and this was done to make sure that after 3 months the bread was not starting again, and this is the case so far. So promising result, I will say. There are more and more wineries that use it. What about the mechanism of action? There are two main hypotheses. It's not completely clarified to date, and the major one is that this chitosan is a powder that gets into the liquid, and it's not completely soluble. So it creates a kind of film, a layer, that then settles, and it gets in contact with the membrane of the wall and the membrane of the cells. It doesn't enter the cells. So it seems that the toxic effect is a kind of response similar to the stress response induced in the cell, but we have no data about which enzymes are involved so far. I hope this will come soon as well. And another idea is that there's a kind of block between the inside and outside environment from the cell, so the compounds can pass through. This is another hypothesis, but the major one is this one. We hope we will demonstrate this very soon. And here again about the action mode, it seems that the chitosan puts bread in a kind of sublethal state for a while. And you have it here. Here you have 10 days after the treatment. This was the cell count, the population before. 10 days after the treatment, I'm sorry, this should be also here. This is my mistake. 10 days after the treatment, you have the plate count that goes down quickly, but on the real-time PCR, you need some time to get the cells completely killed. So it seems that they are the membrane are damaged, but for a while their DNA is still complete, so the real-time PCR will detect them, but after a while also via real-time PCR there's no bread anymore, there's no growth. For sure, what I'm keeping saying, for having long-term results, like 6 months results, you need 6 months, so we'll have them next spring. We don't have them available yet. They are promising results. So here you just have a kind of review of the treatments for bread cells, and about the chitosan, it's less hard to the wine compared to the flash pasteurization. You don't need to filter, because some people don't want to filter, because filtration is very effective as well. It's something completely biodegradable, and it's easy to use. But on the other hand, it needs some time to act, while filtration, once you put it on, it works when you want, and it's not going to be less expensive than the other methods, probably. So that's it. These are the people I have to thank for the work on Brettanomyces at the University of Burgundy before and in Lallemand now. And for sure, thank you for your attention. Okay. So my second presentation, is about another topic, pretty different, and that might interest you as well, because it's related to a region, a terroir in Italy, in northeastern Italy, which is, let's say, our cool climate region, okay, compared to Italian climate. And this is the selection of a wine strain from that terroir, and it might show you the stages of an yeast strain selection in Italy. So the goal now in Europe, and in particular in Italy, is to have strains that really fit a territory and make possible the enhancement of the characteristics of these specific wines. This yeast comes from a wine named Raboso, which is very high tannin, high acidity, very colored, and this is true for the grapes, and it's true for the wines as well. So the wine keeps this kind of characteristics. About the colors and the polyphenols, I just let you know that this was part of my Ph.D. work, so I really know this strain. And when we were to collect the wines in the wineries, we didn't use the glass bottles, because it was too hard to clean. We used the plastic bottles and then discard them. So just to let you know how much the color is in this kind of grape mass, for sure, it was not wine. So this is the wine, pretty challenging for yeast. And this is the project for helping the winemakers of this area. So there was the University of Padova involved in the first stages. Then this is the local winemaker association that really wanted this strain, so they helped a lot, especially for sampling their vineyards and winery trials. And then Lallemand, who was involved from the beginning, because it was the enterprise was interested in such kind of strain. So from where we have this strain? This is the area Veneto in northern Italy, and this is the specific area of Raboso. So it's a very challenging condition for yeast due to the pH, the tannins and anthocyans in this grape variety. So about the sampling, I'm sorry, the pictures come from another selection project, so it's white grape, but it's exactly the same. We collected the samples among the vineyards because we cannot collect masts from the wineries because everybody used selected yeast in this area. So you will only collect the active dried yeast, the same strains as the active dried yeast in the wineries. Then a short fermentation was done in the grape, because otherwise you would have a lot of non-Saccharomyces yeast from the grape itself, while if we waited till 8% alcohol, more or less, on fermentation, and in this way we got much more Saccharomyces that were resistant to ethanol. And then a step of genetic analysis on these strains to make sure they are Saccharomyces and they are different one from each other. And then sensory analysis. What is particular of this selection project is that sensory analysis was very important. So the strains were chosen by their impact on the wine. The first time on very small, we call them nano-vinification, and then scale up to micro and winery scale. So this is the territory. This is the genetic analysis we did. The first one was to make sure to collect only Saccharomyces. Okay, so this is a method, a genetic method, a PCR method. When you have two bands, you have Saccharomyces, that's sure. When you have only one band, which is an internal control, you have other yeast from the vineyard. So instead of working a lot on plate counting, we work a lot on genetic analysis. It's more work, but it's more reliable to the end. So this way we had 239 Saccharomyces on the 700 and something colonies, and then we had to check the strain redundancy. I mean, this 239 Saccharomyces could be only one strain, widespread, or 200 different strains, we didn't know. So again, genetics and the mitochondrial DNA, and we had 130 different strains. And we also checked that there were not commercial strains. And in this vineyards, very far from the winery buildings, we still found 6 commercial strains, all far from the winery. And this has been detected in France as well in another work. So then we went on. First screening was of synthetic mast, the genetic analysis I showed, and also the fermentation performance. This was only to discard the very bad ones, for example, the sulfur smell or something, or strains that could not finish the fermentation. And we ended up with 107 strains that were tested in the natural mast. Again, double-checked the fermentation performance. So here it was a little bit more thorough. And this was the mast condition, so pretty high malic acid and very low pH. And on this small fermentation on natural mast, we did sensory evaluation and some analytical control. So this was the main screening, and we ended up with only the 5 best strains here. About the 5 strains, we have this is their fermentation performance. They were all pretty good, and this one is the one that has been selected finally. It was good in the closing of the alcoholic fermentation. This is kind of a zoom of this part. And also, it was good for malic acid degradation. This is the year after the one I showed, and this was microvinification, so 100 liters, pretty small still. And we started with a lot of malic acid that year. It's not common, but some years it happens. And in this case, the strain could remove a little bit and smooth a little bit the malic acid, which was interesting to the winemakers. Then from the wine tasting, we had, I'm sorry, this is in Italian, I can't translate because I don't have the source file. Here, this is the strain that has been selected. We have some hedonic descriptors. So here you have the pleasantness and the typicity, and it was mapping together. Then we have some floral and fruity aromas. Here you have the cherry, and here there's the blackberry, yes, and some other descriptors like aroma intensity and flavor intensity here. So this one among the 5 was chosen mainly because of this result on sensory aroma, and for the malic acid smoothing also. The fermentation process has been monitored in an online system. So this is the strain, and the orange one was the control, which is a Lallemand VRB strain, and it was more or less the same fermentation kinetics, so it's supposed to be a good fermenter. And so again, other trials, I will be short, on the microvinification and winery scale vinification, again low pH, and this time normal, I would say, malic acid in this area, and the high total acidity, you can see it. Good fermentation performances, again, this was the passito. So you probably know that appassimento is pretty popular in Italy, which is grape drying, and for Raboso they do this as well, so they dry the grapes. And if you go back, it's not very much because sugars go only from 217 to 265, so it's pretty mild. But the strain gave some good results also on this side. In this case, the malic acid was lower, and the strain didn't remove a lot. So it seems to be a strain that can remove a malic acid when it's highly concentrated, but when you have normal to low malic acid levels, it doesn't touch them. What is more interesting is that it was shown to be very bacteria friendly. So this is a malolactic fermentation with inoculated bacteria, and this was done with the same bacteria at the end of alcoholic fermentation with the three strains. and it was the most friendly to malolactic bacteria. And this is interesting to this kind of winemaking because they want the malolactic. Let's see here. We have again some sensory analysis after this second year vinification, and again it was the one that gave most interesting results together with another. And also we kept also the other one, which was here. It was also in the trial before, because there was still one checkpoint, that is the industrial drying of the strains. So Lallemand wanted 2 strains to make sure that if in case the best one was not didn't have a good viability after the drying, they would to test the second one. But it was fine. It was good for this. Here we have another. I think it's the last one. This is a guy in a winery that does natural wines. So no sulfite, no temperature control at all, and usually it doesn't inoculate any yeast. And these are the, this is the final chosen one, this was the other, the implantation test, so how much this yeast can dominate in this kind of situation with high sugar and no control and no sulfite, and it seems to dominate pretty well as also. That's it. Then we have other trials, and this strain is now sold. It has been tested with other top red strains because the goal is to use it also for other winemaking conditions. So it was tested for, this was a Sangiovese trial, and with several other good red strains, and again good results and good malolactic fermentation after bacteria inoculation, so still malolactic friendly. And here you have some chemical parameters, so good for the Sangiovese trial. And again, this was a trial in Spain because this strain has been tested in different countries, and again it gave particular sensory properties to these wines. It was tested with the 71B, that is a very aromatic strain that brings some fermentative aromas, so banana, this kind of aromas. And what turns out is that the aromas provided at this strain are really different, and they seem to be really in accordance to its native area, I will say. The Rebozo, which has this ripe fruit, dark fruit aromas. And it seems that this yeast shows them also in other grape varieties. This again, the malic acid has been checked, and the volatile acidity always pretty low, the same as other strains, more or less. So that's it. This is now these are the an overview on the characteristics of such a strain, and it has a rapid growth, regular speed, good fermentative performances, which were not probably not outstanding, but the aim of this selection project was to find something that is really good for this kind of wine and gives a special touch. So it smoothens the tannic structure, the high acidity seems to make the wines more pleasant, and that's a good synergy with malolactic bacteria. It has been tested also on rosé wines because they do rosé Rebozo as well. So again, this is some pictures from the landscape where we selected the yeast, so maybe some interesting. And this is the winemaker association that really they did want this strain, so you can tell right. And again, some people involved in the project that I have to thank. And thank you for your attention again. This was a trial in which the non-Saccharomyces yeast Torulaspora del Bruchi has been tested for Amarone wine, which is a very particular and peculiar passito wine. I will explain you why the Amarone is. These are the grape varieties that are very local, and it's harvested. And when the Corvina is harvested, it has usually 12, 12.5, 13 maximum potential alcohol percent. It ends with 16, 17 alcohol to define. The weight loss, it depends, but sometimes you end with a 30% weight loss, more or less. And so the grapes are harvested and they are put in such kind of place in open space, because the DOC, which is the quality control for Amarone, doesn't allow a complete control on the drying. So you can't put the grapes in a closed system where you control temperature and acidities. This is forbidden by the law, because it has to be a natural procedure. So the grapes are just put in such places where the windows are open, and what the winemaker do is having some fans working and opening and closing the windows according to the temperature inside and outside, and the humidity for sure during the appassimento procedure. So it's harvested at the beginning of October usually, and at the end of January. Usually they have the good sugar in the grapes. This is for the fermentation. Usually what the winemakers do is that they crush the grapes, they inoculate the yeast, but for a while the temperature is very, very low, kind of 5 degrees, 1 degree, 8 degrees, it depends, because they don't warm very quickly. They leave some days this way, then they warm up a little bit and the fermentation starts. This is really popular in this area. I think it's kind of a tradition. Sometimes after a while they have to inoculate again. There are some winemakers that use different yeast strain. The first ones for the high sugar must, and then after a while they add a high alcohol resistant strain. Not all the people. Some people just use one inoculation. So we started to think about why not using Torulaspora in the first stages, because Torulaspora, the rookie, is known for a low volatile acidity production, very low. So in the first stages, when you have Saccharomyces that can produce some volatile acidity, usually it produces it at the very beginning for the osmotic stress. Why not using Torulaspora instead, which could be safer from this point of view? And then for sure you need Saccharomyces to achieve the fermentation with higher potential. What I didn't say is that Amarone is called Amarone because it's a dry wine, almost dry wine. So it's passito, it's high sugar at the beginning, but it's not a sweet wine at the end. You end up with usually less than 10 grams per liter sugar, so all the sugar is fermented into alcohol, and you need a very strong yeast strain to go so far in the fermentation. So here you have some of the characteristics of Amarone. This is just a commercial product. It goes the appassimento in these bamboo racks. This is the fermentation, then it has to stay 3 years in barrels. So here you have the proportion of barrels. This is this winemaker, but it has to stay, I think, at least 24 months, so at least 2 years, then they can keep it more in barrels. And this is an example of one particular Amarone wine. So this is more or less the product you end up with. These were some results from a grape mast just after the crushing. And what you can see is that the pH is still pretty low. It's not a high pH. And this is due to the Corvina variety. Corvina is somehow, well, if I had to find something a little bit similar, it's Pinot Noir. So it's low color, some tannins, and not very high alcohol potential, and pretty low pH usually. So this was this just to give you a picture of what can be a must of Amarone. Not very high malic acid. So we had some trials. The first one, to make the story a little bit short, was to see to use the Taurulaspora with Saccharomyces. We had a control with only Saccharomyces, one in which the two yeasts were used in sequential inoculation. That means Taurulaspora alone for a while, and then Saccharomyces added, which is the main way of usage of this kind of kit, and the simultaneous inoculation, so both at the very beginning of the vinification. We would like to know what's best for Amarone wines, and this is some results that people from the University of Verona provided me about this is the cell population, so the dark is the Taurulaspora and the other is Saccharomyces, and if you do a sequential, you have for sure all Taurulaspora, so very good in plantation, and then it lowers. But with the sequential, with the simultaneous method, you for sure have a less impact of Taurulaspora because it's never alone. There's Saccharomyces that even in low proportion starts very quickly. These were the results, and what we had was that the winemakers was very good, so it didn't have volatile acidity in the control either. So we didn't see a very better result on volatile acidity. Some people end up here with 1 gram per liter of volatile acidity. In this case, we suppose we will see the difference with Taurulaspora and without Taurulaspora, because in the first stages she is alone, this yeast is alone, and it can lower the volatile acidity. In this kind of trial, the guy was too good, so we just had good volatile acidity in all the trials. What we also had is that this is gas chromatography, and you can see that this is the PCA after a lot of determinations. It seems that the use of Taurulaspora and also the inoculation time has an impact on the aroma. So they group together a little bit, and this ABC were the sequential, DEF were the simultaneous, and this is the control. So Taurulaspora has an impact on the esters, fatty acids, and fusel alcohol concentrations, and it seems that also the inoculation time has an impact. For the sensorial point of view, this was an article on an Italian review, and the sequential was probably the one that was more appreciated by the winemakers in the panels. In any case, you can see that there's an impact, and then after the GC analysis, we expected a kind of impact on the sensory analysis. And other trial, just to see if Taurulaspora needed an acclimatization, because it's a high sugar must. So it's not the same as a normal wine, and Lallemand provides a kit which is ready to use for normal wines. And the researchers at Verona University, they said, okay, but maybe we need an acclimatization because there's high sugar. So there was all was in sequential inoculation in this case. So from the results before, okay, let's go for the sequential. The bee was acclimatized for 48 hours in this kind of mass before getting into the wine, and then Saccharomyces later. And what we saw, it was that for the cell viability, that didn't make a very big difference from the trial before. And from the analytical point of view, we had again more or less the same. And acetic acid here, we had lower in the control. Malic acid, this was a little bit, this was a first trial. We don't know if it will be confirmed, that it seems that Taura is kind of malolactic friendly. So it seems that it favors the malolactic fermentation, probably because there's a long lag phase at the beginning. Torulaspora is not as quick as Saccharomyces, so the first stages of fermentation are a little bit slower. So this is not confirmed yet, but in case you want to use Torula and avoid malolactic fermentation, be careful, follow, and then sulfite. If you want the malolactic fermentation done, probably using the Torulaspora, they will be even easier. And in this case, the TD, the Torulaspora, had an impact, you can see it, but the inoculation time, the pre-acclimatization doesn't have any impact. So it seems that it can work well, good cell viability, the same with or without acclimatization, and it doesn't have an impact. But what was confirmed is that the use of Torulaspora still maps to the wines map together, and that's it. I'm sorry, it's just a small part of the research, but it's not my field, my personal field.
Thank you, Tiziana. You've shared with us so much information on 3 really relevant topics, and we're just so lucky. She was just speaking earlier this week at a conference in the Finger Lakes. So just through happenstance and our connections with Lallemand and Scott Labs, we were able to bring Tiziana here to be able to share her expertise with us. We have the room till 12:30, so we have lots of time now for some discussion, and I'm sure with all the information that you've shared with us, there's going to be some questions from the audience. So I open up the floor now and see first if there's some willing partakers to discuss some issues with Tiziana.
Going back to the chitosan treatment, are there any implications for filterability?
Well, it seems that no, because you need to rack off after this treatment, and this kind of layer goes down. So after racking, you don't have any more chitosan or really traces. So you definitely need to rack off before going to filtration, that's sure. But it's something we give in the protocol. Also, what's to date, the protocol is 10 days treatment and then rack off. We are now having some trials also in Italy to try to let the product stay with the wine for up to 6 months, because some winemakers, they don't want to rack the wine, especially if it's in barrel, that they don't have so many barrels. And we will make sure that the product is still effective, and for sure, and principally that it doesn't affect the wine color, stability, tannic structure, whatever. And then probably it will, the protocol could be changed a little bit wider. But in any case, before going to filtration, you need to rack off. And since it's a kind of film, you can eliminate it mostly, 100%.
And so the racking losses are very minimal.
Yeah.
Judge.
Will these chitosan treatments, will they done in barrels or in tanks?
Both. We have some trials in tanks. We have some trials in barrels. I would say the barrels they are coming now. So there is the result I showed you. There were 1 in barrel and 2 or 3 were in tanks from last years. In Italy this year we have some wineries that are doing trials in many barrels as well.
As a follow-up question, do you think that the chitosan will inhibit the reoccurrence of barrel-infected tanks?
Oh, that will be great, but I don't think so. So there's a kind of parallel research or interest in searching if it can be useful for treating the barrels. No information to date. What is difficult from this point of view is that it doesn't get into the solution. So it's hard to think that it will go into the very narrow place. But I have no information, so that would be, yes. So after the treatment, the wine is racked off, but we cannot assure that the barrel after you have to clean with other systems. It's not clean the barrel.
Linda, did you have a question?
Actually, those were my two top questions, but
Yeah.
my other question was, what's the particle size of the kind of sand? Is it a health risk for handling in the winery?
No, nothing. No, it's really a sugar. It's a polysaccharide.
Yeah.
You think something like paper or starch. Starch is something that yes, agarose, this kind of compounds. So it's really friendly. Yes, nothing.
And you had mentioned that it's used in the food industry.
Yes, the food industry is already used, and in some healthcare products as well. So it's really, and also it doesn't come from jellyfish, so no issues for allergies and like this.
I guess I was wondering, did you mention that in Italy for the passamento process, when you
Yes.
started the fermentation, you sometimes started at a cooler temperature?
Yes, sometimes some winemakers. Yes.
So and is the logic behind that to sort of slow down the yeast growth rate and just maybe?
Well, no,
get
it doesn't.
used to
It
that?
just comes from the fact that the grapes are very cold at that point. Okay, so it's January. You have very cold. It's not a nice wine, but you have pretty cold temperature around, and the harvest is can be at 5 degrees or 10 degrees. So I think they possibly want to avoid a very dramatic change of the temperature, and this is why they did it. There's not nothing more, and it's this kind of low temperature is room temperature. I mean, it's not cool down, but it's the winery temperature at that time. They just wait a little bit before start warming it. There's some winemakers, not all of them.
I can imagine it may also just be because of this is an older traditional method, and they may not have had initially ways to heat up the fermentation.
Yes.
So this was just the tradition that started, and has
Yes,
been.
I think so. That they try to keep the tradition as much as possible. Yes.
For the Rebozo yeast strains,
Yes.
maybe I missed it. I saw the fermentation temperature was 27 degrees under the cap. Now, is there what is the fermentation range, and what would be the optimal?
I think it's from 15 to 28. I will
15 to 20.
38, yeah, something. I have to double check this. I'm sorry because I can't remember. But yes, it has not been tested very much on white and rosé wines, but we are now trying it on white and rosé wines. So the lower limit might be decreased, or probably I have to check on the data sheet. It has already been kind of little bit decreased. The one we had was really for the red winemaking, normal red winemaker with or without temperature control. So it was not really. It's a strain that doesn't really care that much about the temperature changes during the fermentation.
Just to follow up on that particular project, because I see some students that are here in the lecture today. Can you give us a sense of the time that it took in terms of years to go from the initial selection through to now having a commercial product? Just to put some context.
It started in 2005. So 2005 and 2006 was grape picking and grape selection. 2007 was the very small scale vinifications mostly, and between 2005 and 2007 all the genetic analysis and the lab scale work. 2007 we had the first 100 liter fermenters on the 5 strains. 208 and 209 we had the 3 strains to be checked again. So other small scale fermentation all in the Rebozo area. So this was 2008 and 2009. Also winery scale. So the first winery scale trials were in 2009. And then 2010 was mostly Lallemand trials on other varieties, other kind of juices with different conditions in different areas of Italy and of Europe. And we ended up with the product on the market in 2011. And you have also, at one point, I think it was in 2009, there was the industrial production test for sure of this. So it has been kind of 6 years, yes. It's possible that it can be a little bit quicker, but I think not that much. Maybe one less year sometimes, but it takes some time. Yeah. I don't know if here you have different timing experience.
No, I think that's a good outline of the length of time to go from an initial idea and some initial selections through to commercial product. Any other questions from the group? Okay, well, please join me again in thanking Tiziana for her time this morning, and Matthias Voss as well.